In this video, we will discuss about the first class of lipids called triglycerides. Triglycerides are also known as neutral fats, also abbreviated as TAG, also called triglycerol. These molecules are made up of two components: fatty acid and glycerol. Fatty acid is an acid and glycerol is a sugar alcohol. Both of these molecules, fatty acids and glycerol, they can either be biosynthesized or they can be eaten or ingested via diet. So both of them can be either synthesized or they can be taken via diet. In the last video, we discussed the structure of a triglyceride, which was there is a glycerol molecule which has only 3 sites available for the fatty acid to attach. Fatty acid comes and attaches via a special linkage called ester linkage. And therefore, triglycerides are also called fatty acid esters of glycerol. What we are going to do in today's video is that we are going to look at both these components, fatty acids as well as glycerol, and at the end of the video we are going to join both of them to form simple lipid category 1 triglycerides. Let's discuss fatty acids first. What are fatty acids? They are acids and therefore they are going to have the acidic group, the COOH group. The COOH group or the acid group is attached to different carbons, and in fatty acid there can be 4 carbons till 36 carbons. So they can be short chain fatty acids as well as long chain fatty acids. Again, depending upon the bonding between these carbons, fatty acids are divided into 2 categories. If there is no double bond present, then that fatty acid is called saturated fatty acid. But if double bonds are present, the fatty acid is called unsaturated fatty acid. So let's have a look at structure of saturated and unsaturated fatty acids. As we said, that fatty acids will have a COOH group. So let's take that group. This is attached to carbons. Let's make an imaginary saturated fatty acid composed of 6 carbons. 3, 4, 5, 6. 4, 3, 4, 5, 2, 6, 2. So this is a saturated fatty acid. When we are representing the saturated fatty acid, this fatty acid appears linear. So we can draw the saturated fatty acid as, so carbon number 1, 2, 3, 4, 5, and so this is the structure of this particular saturated fatty acid. If we line up more fatty acids along with this particular fatty acid, again the same type of fatty acid, 6 carbons, we can see that this structure is organized and it is in order. So it has some order. So these particular fatty acids, which do not have any double bonds between the carbon in them, they are more packaged and have more stability. Therefore, the kind of fat which will have more saturated fatty acids in them, they will require more heat for them to melt. Therefore, we say that these saturated fatty acids have a higher melting point. Okay, let's move on to unsaturated fatty acids. Unsaturated fatty acids will have some unsaturation in them. Let's make one imaginary unsaturated fatty acid again with 6 carbons. This particular unsaturated fatty acid again has 6 carbons, but there is unsaturation present between the fourth and the fifth carbon. Now, if we have to draw this particular unsaturated fatty acid, what we'll get is structure unsaturation. If we compare this structure with this structure, you see that there's a little bit difference. And this difference arises because of the presence of this particular double bond. The presence of this double bond here in this unsaturated fatty acid gives a kink in the structure. So the structure is not linear, but it is a bent structure. So now, if we line up some unsaturated fatty acids right next to this particular unsaturated fatty acid, what we'll get is that there's going to be a little bit more disorder. This structure is disordered, and because it is disordered, it will have less packaging, and as a result of that, the melting point of this particular unsaturated fatty acid will be low, which means that less amount of heat will be required to melt this particular fat having unsaturated fatty acid. Now, because of the presence of the double bond, the unsaturated fatty acid can have two different types of structure. Those two different types of structure are, they can either have a cis structure, cis unsaturated fatty acid, or a trans structure. In cis unsaturated fatty acid, the position of hydrogen around the carbons which have the double bond will be on the same side, whereas in trans unsaturated fatty acid, the position of hydrogen will be on opposite side of the chain. Cis unsaturated fatty acids are present in nature, so they are more prevalent, whereas the trans unsaturated fatty acid are formed as a result of fermentation and hydrogenation. And these particular trans unsaturated fatty acids, they are unhealthy and they are known to cause increase in LDL. They increase LDL in the blood. And this LDL is low density lipoprotein, which is healthy. Unsaturation also results in two different kinds of unsaturated fatty acids called monounsaturated fatty acid, monounsaturated fatty acid, also labeled as MUFA, or polyunsaturated fatty acid, also called PUFA. Monounsaturated fatty acid will have a single unsaturation, like this. This is an imaginary MUFA, and polyunsaturated fatty acid can have two or more than two unsaturation present. Okay, so these are the different variations of unsaturated fatty acid which can be found in nature. Now let's have a look at few examples of different types of fatty acids. Most common example is myristic acid. And this myristic acid has 14 carbons in it. So myristic acid has 14 carbons, but it does not have any unsaturation. So how do we denote myristic acid in sense that we need to know how many carbons are there and how much unsaturation is there? We denote it like this. So let's draw myristic acid. It has 14 carbons. So this end will be called carboxy end, and this end will be called ethyl end. So this is myristic acid, and the type of acid this is a saturated acid, and it has no unsaturation. Moving on to next example, which has 16 carbons, palmitic acid. Again, it is a saturated fatty acid. It has 16 carbons and no unsaturation. Another example is stearic acid. It has 18 carbons and no unsaturation. So if you note, mostly there are even numbers or even number of carbons present in fatty acids. So in nature, usually even number of carbons are present in fatty acids. These are a few examples of saturated fatty acids. Let's take up unsaturated fatty acids. First example is palmitoleic. Palmitoleic acid is derived from palmitic acid, therefore it is going to have 18 carbons. But it has one unsaturation. So at carbon number 9 and carbon number 10, there's an unsaturation present. So how do we know denote that there is unsaturation present at this particular carbon number? We'll denote it by using this. What this whole formula is telling us is that there are 16 carbons. There is one double bond, and this double bond is present at the 9th position. So this is palmitoleic acid. Another example is folic acid. Folic acid, again, like stearic acid, has 18 carbons and only one unsaturation. So how do we denote the unsaturation? We denote it using this delta nomenclature, where it is telling that at carbon number 9 in folic acid, there's going to be unsaturation. All these are unsaturated fatty acids. Then after folic acid, we have Next example, linoleic acid. Linoleic acid is derived from oleic acid, has 18 carbons, but it has 2 unsaturations. At which position? At position number 9 and position number 12. Next example is linolenic acid. Again, it has 18 carbons, but it has 3 unsaturations at carbon number 9, 12, and 15. So there's a pattern here where unsaturation begins at carbon number 9 and moves to carbon number 12 and then carbon number 15. There's one special case of unsaturation in arachidonic acid. Arachidonic acid has 20 carbons and 4 unsaturations, but the unsaturation is present at different positions. It is at carbon number 5, 8, 7, and 14. So these are a few examples of unsaturated fatty acid. So now let's have a look at the number of unsaturation and categorize them under MUFA or PUFA. Palmitoleic acid is PUFA, oleic acid again, PUFA, and the rest of these are PUFA. And out of the last 3, linoleic acid and linolenic acid, these two, they are essential fatty acids, which means these two cannot be synthesized in a body. We have to take it only from diet. There are also omega fatty acids. What are omega fatty acids? Let's take example of linoleic acid. This linoleic acid has 18 carbons. So let's draw linoleic acid. In case of omega fatty acid, what scientists did was that they started counting from the methyl end. Okay, so when we are counting carbons from the carboxyl end, we say that the nomenclature is delta nomenclature. When we are counting from the methyl end, we say that it is omega nomenclature. So when we start counting carbon numbers from the methyl end, we see that the first unsaturation is present at carbon number 6. So we say that linoleic acid is omega 6 fatty acid. There are cases where unsaturation might begin at carbon number 3 and that fatty acid is called omega 3. Unsaturation might also begin at carbon number 9 and they are called omega 9 fatty acids. So this is omega nomenclature. Coming back to this list, let's now include omega nomenclature in this as well. The linoleic acid is also omega 6 acid. So we'll write it as omega 6. Linolenic acid is omega 3 acid. Omega 3 acid. Whereas Folic acid is omega-9. So omega acids are denoted by N. So N3, N6, and N9 omega acids are linolenic acid, linoleic acid, and folic acid respectively. Let's move on to the next component, which is glycerol. Glycerol is a sugar alcohol having 3 carbons, and each of these carbons have OH group attached to it. So this is glycerol molecule. Again, glycerol molecule can be biosynthesized in the body, or it can be taken in via food. So what do we have now? We have fatty acids, we have glycerol. So what we can do is we can join both of them together to produce a triacyl glycerol. A simpler way of looking at synthesis of triacyl glycerol is joining glycerol with different fatty acids or the same type of fatty acids. Let's look at a simplified version first. That's some fatty acid. Let's take some imaginary fatty acid having 4 or maybe 6 carbons, and we take 3 molecules of this. So we'll join these 3 molecules of fatty acids together. While joining these, what happens is 1 molecule of water is going to be removed. This OH and this H of glycerol. These 3 molecules of water will be removed. As a result of this, what we are going to get? We are going to get this is glycerol. And this bond bond. Now, if we have a look at this particular triacyl glycerol molecule, it has the same type of fatty acid, having the same number of carbons and having no unsaturation. So, you'll say that this triacyl glycerol is a simple TAG. If another fatty acid, another type of fatty acid comes and attaches here, then we'll say that if all the fatty acids are different, we will label this particular triacyl glycerol as a mixed triacyl glycerol. Usually how the synthesis occurs is glycerol plus 1 fatty acid result in formation of monoacyl glycerol. Monoacyl glycerol plus 1 fatty acid result in diacyl glycerol, and diacyl glycerol again plus 1 fatty acid will result in triacyl glycerol. These fatty acids can be same, these fatty acids can be different, resulting in formation of either simple triacyl glycerol or a mixed triacyl glycerol. This is a simplified way of formation of ester bonds between glycerol and fatty acids. How does the biosynthesis actually occur is a very different process. Triacyl glycerol biosynthesis in our body occurs through many different pathways. The most common pathway is the Kennedy pathway, also called glycerol 3-phosphate pathway. And this pathway is undertaken in liver cells and adipocytes. Let's have a look at this pathway. Through glycolysis, glucose is converted into glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate gets converted into glycerol 3-phosphate by the action of dehydrogenase enzyme. Now this glycerol 3-phosphate will be used or will be acted upon by another enzyme called acyl transferase. And this acyl transferase is going to transfer one fatty acyl group attached via coenzyme A onto glycerol 3-phosphate. So now this glycerol 3-phosphate will have one fatty acid attached to it. If you look at structure of glycerol, we know glycerol has 3 positions. At the 3rd position, phosphate group is attached in glycerol 3-phosphate. When acyl transferase acts on this glycerol 3-phosphate, one fatty acid group will attach to this glycerol 3-phosphate to form lysophosphatidic acid. And the structure of this acid is going to be like this. There's a phosphate group attached, OH group, OH group. At the first position, fatty acid will be attached. Here, coenzyme A will be released. Now, this particular molecule, lysophosphatidic acid, has one fatty acid, one OH group, and one phosphate group attached. So, the next step is addition of one more fatty acid in the second position. So, another fatty acid will come, and by the action of acyl transferase, it will attach to the lysophosphatidic acid to form phosphatidic acid. And let's have a look at the structure of phosphatidic acid. Phosphatidic acid will have phosphate group, fatty acid attached at the second position, and fatty acid attached at the first position. The only thing now left is to remove this particular phosphate group. This phosphate group is removed by the action of phosphatase enzyme. And now this phosphatidic acid will be converted into 1,2-diacyl glycerol. And the structure will be like this. Acid 1 and fatty acid 2, and here OH group will be present on the third position. This 1,2-diacyl glycerol just needs one more molecule of fatty acid. So what is going to happen? Third fatty acid is going to come, and by the action of another acyl transferase, it is going to get converted into triacyl glycerol, which will have fatty acid 1, fatty acid 2, and fatty acid 3. This is how a triacyl glycerol is synthesized by Kennedy pathway in liver and adipocytes. Another pathway or second pathway is dihydroxyacetone pathway. In dihydroxyacetone pathway, first gets converted into 1-acyl dihydroxyacetone phosphate again with the help of acyl transferase. And this one acyl dihydroxyacetone phosphate will get converted into lysophosphatidic acid with the help of oxidoreductase. So this is the second pathway, which is called the HAP pathway, and it occurs in peroxisomes and endoplasmic reticulum. There is a third pathway called monoacyl glycerol pathway, and this takes place in the intestine. Here, what is happening? Fats are being acted upon by lipase to form fatty acids and monoacyl glycerol. This monoacyl glycerol will be acted upon by acyl transferase, and this acyl transferase is going to transfer one fatty acyl group to the glycerol molecule, and that will result in attachment of two fatty acid to the glycerol molecule, resulting in the formation of 1,2-diacyl glycerol. This diacyl glycerol will get converted into triacyl glycerol with the help of another acyl transferase that will attach one more fatty acid to it. There is one more pathway, fourth pathway, where triacyl glycerol is being synthesized from two diacyl glycerol molecules. So, diacyl glycerol plus diacyl glycerol. Remember that diacyl glycerol has two fatty acids attached to it. So, this will get converted into triacyl glycerol with the help of an enzyme which is going to transfer one fatty acid group from either of these diacyl glycerol to the initial diacyl glycerol. And this enzyme is transacylase, which is a type of transferase. What it does is fatty acid 1, fatty acid 2, which fatty acid 3, fatty acid 4, which. So these are two diacyl glycerol. What the transacylase will do, it will transfer one fatty acid to this position, resulting in the formation of TAG, and what we are left with is one monoacyl glycerol. And this fourth pathway is called diacyl glycerol pathway. And this occurs in intestine and seeds. So this was all about formation of triacyl glycerol from different pathways. In the next video, we are going to talk about the next class of lipids, which are conjugated or compound lipids.